Thank you very much, Nori, for agreeing to give a Build a Cell seminar, and you can get started.
Okay. Thank you very much, Kate, and thank you for inviting me to this Build a Cell seminar. I'm Norikazu ICHIHASHI from the University of Tokyo. Our laboratory are doing in vitro synthetic biology, bottom-up synthetic biology. Today, I'd like to talk about our recent project. We have two research objects. Both are based on bottom-up synthetic biology. First one is to understand how Darwinian evolution produces diversity and complexity. And second one is to design and realize a regenerative molecule system in a test tube. So today I'd like to show you our recent progress in this part. I have a chat. Oh, yes, thank you very much. There's a question in the chat. The first page is AI drawings. I asked draw a picture based on artificial cell. So I don't understand what it is, what this is, but I think it's beautiful and I love this. Okay. So here I show you a basic scenario towards the origin of life. So there is a part here, okay. And after the foundation of the Earth, it is believed that prebiotic chemistry began. I'm sorry, okay. There is I'd like to use pointer. Wait a moment. Okay, here. After foundation birth, people believe that prebiotic chemistry began, and then RNA world began later. And then the DNA and protein life appeared, and they diversified into present-day very complex and sophisticated living things. And most important event occurred, I think, around here. That is the Darwinian evolution. Darwinian evolution would started probably before RNA world, and then the ancient living systems developed their complexity toward the complex present-day living systems. So this is the basic scenario. People believe that somehow Darwinian evolution produced present-day complex living things, but we are curious. We have a question: how does complexity develop through evolutions? We have this question because we are doing in vitro evolution. We are doing evolutionary experiment in a test tube, and we found that no complexity develops through evolution in a test tube. Usually, evolution easily, we can realize the evolution in a test tube, but molecular systems usually doesn't increase their complexity, but usually it gets simplified. So our question is, how does complexity develop through evolution? To answer this question, our strategy is very simple. We try to construct an evolvable replication system and perform experimental evolution and find a condition that allows development of complexity. So, but we need a hypothesis how complexity develops in a molecular system. Then here is our hypothesis. This hypothetical pathway is based on the idea or theory by Maynard Smith and Satomari. And they said division of labor and cooperation increase the complexity of living things, living systems. So based on this theory, we think that if this happens in the molecular systems, then a molecular system can develop complexity. In our hypothesis is like this. First, we start from a simple replicator, and they diversify into two different replicators, which have a little different functions, and then they start to cooperate to form an interdependent replication network, and then finally fused into a single RNA molecular system. In this way, the first simple replicator, which have only one function and only one gene, developed into a more complex replicator, which have two different genes and functions. So we'd like to see actually this process happens in a test tube. So to do so, first we have constructed an evolvable RNA replication system. This is based on the Spiegelman's RNA replication system, but the different point is we introduced a translation system, translation coupled RNA replication system. We use the genomic RNA, artificial genomic RNA, that is about 2,000 nucleotides. And this RNA encodes only one gene, RNA replicase gene. More precisely, it's encode the catalytic subunit of RNA replicase gene, Q beta replicase. And the other subunits are included in a translation system. Then another important factor is a cell-free translation system. This is not a extract of a cell, but it's a reconstituted system, consists of all purified molecules, purified protein and ribosome, and tRNA and amino acid and TP and so on. It is known as a pure system. Okay? So we use a pure system, and we incubated artificial genomic RNA in the pure system. Then RNA replicase is translated. Then translated replicase replicates the RNA, and mutations are introduced during this step. So what we need is to continue this RNA replication for many generations, and then evolution should occur. But to allow the evolution, this system needs another factor. It's a droplet. Compartmentalization is needed to make a linkage between RNA and translated replicase. Indeed, this is necessary, and the compartmentalization is required for this system because in this system, the genotype RNA is a different molecule from the phenotype replicase. So to make a linkage between phenotype and genotype, we need a compartmentalization. Then as a compartment, we use water in oil emulsion, which is a droplet, about a bacteria-sized droplet. It can be prepared easily, just mixing reaction mixture, pure system and RNA with oil and some detergents. Then we encapsulate the reaction inside the droplet and then incubate the droplet. Inside the droplet, RNA replication occurs, translation and RNA replication occurs, and then the RNA replication stopped because of the all NTPs are consumed. Then to continue replication, we diluted the sum of the droplet with a new droplet, which contains a new pure system. Then vigorously mix to induce the fusion and the division of the droplet to supply nutrients. And then again, next round of replication occurs. So through this serial dilution experiment, we performed a long-term RNA replication experiments. Then we found that we continued this serial dilution cycle for more than 100 times. Then we found that RNA replication ability of the RNA population increased gradually. And finally, it stopped. And also mutations are accumulated in the RNA. And finally, the mutation rate became slowed down. So this increase of the RNA replication ability and the number of mutations indicates that Darwinian evolution occurred in our system. But that evolution is not what we expected, because here I draw a phylogenetic tree of the RNA species that appeared during the evolutionary process. First, ancestral RNA is here, and the final RNA is here. So the phylogenetic tree is very straightforward. There is no large divisions. That means diversity didn't appear. So, and also we found that RNA replication scheme didn't change during this evolution. First RNA encode one gene and replicate very simple scheme, but the final RNA also encode one genes and replication scheme didn't change. So this means that evolution actually happened, but that evolution doesn't produce genetic diversity, and also the evolution almost stopped after 100 rounds. So I think that this evolution of RNA replicators is significantly different from the evolution of living things. Evolution of living things produces many diversity and also complexity. So we'd like to know what is difference between evolution of RNA self-replication system in our model and evolution of living things. I think that one of the missing factor in our system might be parasite. We thought that because parasite is believed to drive host evolutions. It is the hypothetical Red Queen hypothesis. It is known as a Red Queen hypothesis. This hypothesis says coevolution with parasite could accelerate host evolution and induce diversification through evolutionary arms race. According to this hypothesis, if we introduce parasite in our system, that drives host evolutions, and that drives a continuous host evolution, and also induce diversity. Okay, sorry, I have a chat. Oh, I have a question in the chat. Box in the RNA evolution experiment, did you see any influence of RNA stability? Was RNA hydrolyzing at all during the experiment? We didn't check it, but actually, yes, it is. It is degraded during incubation, but the degradation speed is slower than the replication speed. So as a net RNA increase, the RNA degradation occurred because very small amount of RNA degradation enzymes are contaminated in the pure system. So RNA, we cannot avoid the RNA degradation. Okay, so let's go to next slide. So we'd like to introduce parasite in our system. And we noticed that in our system, parasite spontaneously appears. In the previous evolutionary experiment, we didn't allow the appearance of parasite because we kept the RNA concentration as low as possible at this level. We can adjust the dilution rate every time, and we can keep the RNA concentration to a low level. But if we didn't adjust the dilution rate, but we performed a serial dilution experiment at a constant dilution rate, result was like this. First, RNA concentration increase, and then at around this point, another type of RNA spontaneously appears. This RNA is very small RNAs, and later study we found that this small RNA spontaneously appeared from genomic RNA through RNA recombination, through a deletion of the internal sequence, internal replicase gene sequence. So this small RNA does not encode replicase gene, so it cannot produce replicase. Then this small RNA cannot replicate by itself, but it can replicate if it is in the same compartment with other genomic RNA, using the replicase produced by the genomic RNA. In this sense, this small RNA is a parasite, and this original RNA, original genomic RNA, is a host. So now we get a parasite. Then we try to another serial dilution experiment in the presence of parasite. So this experiment started from one of the evolved RNAs. So this, without parasite conditions, this RNA barely accumulate more mutations. But in the presence of parasite, the number of accumulated mutation is very high. So evolution didn't stop in the presence of parasite. And we also found that in the presence of parasite also evolved. New type of parasite appeared in the later stage. Then we'd like to check this continuous evolution actually occurred through evolutionary arms race between host and parasite. To test this, we collected some RNA clone at this point, 3 host species and 3 parasite species throughout the evolutionary process, and checks the strengths of each RNAs by competition experiment. Oh, we have a chat. Yes, I have a chat from, guess fluctuating environment would be a key to the generation of complexity. The same environment would lead to non-complex in terms of entropy point of view. Oh, yes, that's a possible idea, and I agree with you. But I think that the environment, in the presence of parasite can change the environment. Parasite is a part of the environment. So in the presence of parasite, I think that environment for host species continuously changing. So that would be a key to allow the evolution complexity. Okay, so here in this slide, we perform the competition experiment of the parasite and host species. Oh, doesn't work, okay. Then we found that, I'd like to skip this figure briefly, but we found that initial host species is weak to the initial parasite, but the next type of host species that appeared during the evolutionary cycle became resistant to the initial parasite. And then next parasite again can infect the next type of host, and again the final parasite became resistant, and the final host became resistant, and the final parasite again infect the final host. In this way, we found that evolutionary arms race occurred in our system. And then we further continued the serial dilution cycle. And next, we'd like to know the diversification occurred, whether diversification occurred in our system. To check this, we performed the deep sequencing analysis at this point, and the results were shown as a phylogenetic tree. This phylogenetic tree is significantly different from the previous phylogenetic tree that was very straightforward. But in this case, phylogenetic tree have a very large branches. So that means diversification occurred here. And the final population, there are 5 lineages in the final populations that is encircled in here. These 5 circles means these RNA existed in the final populations. And ancestor is here. So that means ancestral RNA divided at this point into 2 large branches. This green branch is host RNA. This, and also the size of the length of the branch means the number of mutations accumulated. So this host RNA accumulated many mutations. It evolved very fast. And another branch is also host RNA, and they existed in the final population, but these branches have a short length. That means the evolution is very slow. We don't know the reason of the difference of the evolutionary speed, but this slow evolving host species still survived in the final populations. And another small branch derived from here, this is also host RNA. And at this point, another branch derived from here. This is a parasite RNA. This pink line is a parasite, and the parasite's lineage also accumulated many mutations. And here, at least 2 lineages existed in the final populations. Now we found that in the final population, at least 5 lineages existed. And next, we test the biochemical property of the 5 lineages. Here, the result was shown as schematic drawings. Each circle represents each RNA. these 3 RNAs are host species, and the bottom 2 RNAs are parasite species. And they have very different biochemical properties. So what we found is here, for example, here, HL1, which is a host lineage 1, replicate itself and also replicate one parasite species, PL3. And interestingly, this RNA cannot replicate the other 3 RNA. We tested the replication, but it cannot replicate the other 3 RNAs. So this is a very strict RNA replicator. And in contrast, this RNA, HL2, replicate all the other RNAs. It's prefer this parasite, but it replicate all the other RNAs. So this is a very, very generous RNA. So, and interestingly, these 5 RNAs have a different properties, but as a whole, they replicate very stably. So I think that these 5 RNA species form the interdependent replication network. To test, next we test the interdependency by removing one of the RNAs. This is a simulation result, but based on the experimentally measured parameters. Then in this analysis, we removed one of the RNAs. For example, here, PL2, this is a parasite RNA. If you remove a parasite RNA, then we found that another RNA, in this case, HL1, this host species disappear. This result shows the result. If you remove PL2, the green one, HL1, also disappear from them and cannot replicate. So this means HL1 and PL2 are co-replicated. They are interdependent. That is, and we also found that if you remove PL2, HL2 also disappear, and PL2 also disappears. So these 4 RNAs are, I don't understand, but HL3 cannot replicate in our experiment, but these 4 RNA are replicated interdependently. Okay, here is a short summary of the first topics. What we found is without parasite condition, evolution was very straightforward and the evolutionary scheme didn't change. But in the presence of parasite, the evolution was significantly different. RNA species divided into several lineages and replication scheme became more complex. So I'd like to compare our result with the initial hypothetical pathway. What we found is diversification occurred, and we found that not cooperation, but interdependent replication occurs. We cannot say it's cooperation because there is no direct mutualism, but it's a very indirect interdependency. So, but I hope that if you repeat the evolution experiment for more generations, the interdependency would increase and at some point cooperation may be appear. And finally, they may fuse into a single RNA. Okay, now we are continuing. We are continuing this evolutionary experiment. Okay, I have a chat again. Oh, thank you very much. Interdependency. Thank you very much. Okay, so I'd like to move on to the next topics. To design and realize a regenerative molecular system in a test tube. I have another chat. All these continuous replication experiment in vitro. Yes, all the experiment I show you in vitro. No living thing, the speed don't use. Okay. And the next question is, the question would be, if you repeat the same experiment, would you get a similar result? Yes, it's a very good point. Actually, we did 3 independent experiments, but it's not completely independent because in the second experiment, we started from around here. The initial part is the same, but at this point, we divided into 3 experiments and then perform the same experiment. And I have our data here. The result is almost same. In the second experiment, yes. These are the phylogenetic tree of the second and the third experiment. They are very similar. They divided into several lineages. So I think that this division of the into a large branch is reproducible. Okay. Another question, like matrix starts the world again. Yes, exactly. Yes. Okay, next question. So the diversity is all dependent on replication rather than splicing. Diversity is dependent on replication rather than splicing. Yes, I think, yes, it's based on replication. Splicing, what I'm not sure the meaning of the splicing. Yes, we think replication is a basic event because we use the replication enzyme. So, Q beta replicase, which is the activity of the replicase, is to produce complementary strands. So, I believe that this is a basic process what happened here. Okay. Okay, so let's move on to next. So next topics is to design and realize regenerative molecular system in a test tube. So we'd like to make a regenerative system for bioproduction. Okay. Presently, people for bioproduction, people uses natural microorganisms such as bacteria and yeast. But there is a limitation for these natural organisms in the point of controllability. You may know that if we design the genetic network or metabolic network in the cell, usually the expected design doesn't work because of the complexity of the internal natural system. So to overcome this difficulty, one of the methods is to construct a simple regenerative molecular system bottom up. If we can make a simpler molecular system, that can be more controllable, I think. So based on this idea, we are trying to regenerative replication system. Regeneration is, I think, important to reduce the cost. So we'd like to make a regenerative molecular system, and regeneration need three reactions: DNA replication, transcription, and translation. For regeneration, of course, DNA should be replicated. And for replication, we need replication enzymes. That would be a protein. So these proteins should be encoded into DNA and should be expressed through transcription and translation. And also we need that all the proteins required for transcription and translation, and we have to encode all the proteins into DNA. So we need many genes, and we have to encode every gene into DNA and express every genes to a sufficient amount. So that is what we have to do. So, but if we construct such a system, then in this system, all the macromolecules, polymers, regenerate itself if we supply only low molecular weight compounds such as amino acid and NTPs. So before we started this project, the transcription and translation have been reconstituted in vitro. That is a pure system we used in the previous topics. Then we tried to couple the DNA replication with a pure system. Then, so DNA replication coupling means encode all the genes required for DNA replication into DNA and express in the pure system. And then using expressed proteins, DNA should be replicated. We try to this DNA replication system. So today I'll show you a result about DNA coupling in the pure system and also DNA replication coupling in the pure system. And also we'd like to show you the result about the tRNA expression in a system, in pure system, and 20 ARS, which is translational factors, these expression in a pure system. Okay, first DNA replication. We had to choose which DNA replication scheme we use. There are many DNA replication have been reconstituted in vitro. The most famous one would be a E. coli DNA replication system. This is very efficient replication system that replicate very long DNA. But problem is it requires 20 genes. So present pure system, 20 gene expression is still a challenge. So we had to more simpler DNA replication system. As a simpler DNA replication, phage DNA replication system have been reported. For example, here, 529 DNA replication system need only 4 genes. Recently, the Chris Daniel's group have been reconstituted this DNA replication system in a pure system. But there is still a problem in this DNA replication system because 2 of the 4 genes need at very high concentrations. SSB and DSB need micromolar order. So micromolar order expression is still difficult in a pure system. It's possible, but it's still a challenge in the pure system. So we took another strategy using an artificial DNA replication scheme, which need only 2 genes. This scheme have been reported in 2006 by Forster and Church. This scheme is very simple. Started from a circular DNA that encodes 2 genes, which is 529 DNA polymerase and Cre recombinase. Both are phage proteins, but from different phage. So these 2 proteins can be expressed in the pure system. And then 529 polymerase start rolling circular replication from a circular DNA to produce very long DNA. And then Cre recombinase catalyze the homologous recombination to produce a circular DNA. Through this process, circular DNA self-replicate. But this process, this scheme didn't work at first because Cre recombinase significantly inhibit the DNA replication, DNA polymerization. So first we tried to obtain, we did a directed evolution of the DNA and 529 polymerase that worked in the presence of Cre recombinase. And to make a long story short, we finally obtained the DNA and 5 polymerase mutant that can replicate in the presence of Cre recombinase and to replicate the circular DNA. Circular DNA have different topologies and shows a multiple band, and the finally obtained DNA can show the replication of multiple bands. That means circular DNA are replicated. So now we constructed a DNA replication that coupled with a transcription translation. Then we continue the DNA replication for many generations in the droplet. This through a serial dilution cycle, I'll show you in the previous topics. Then in this case, again, DNA replication continued many generations until 60 rounds, which means 60 days, 2 months. Then as a middle point, we cloned the DNA species and compared the DNA replication abilities, and we found that these DNAs in the round 30 shows higher DNA replication abilities compared to original DNA. And we also found many mutations accumulated in the DNA populations. So these data shows that Darwinian evolution happened. But here, in these topics, our focus is not evolution, but I'd like to show you by this data, DNA replication. Gene expression coupled DNA replication is continuous. And now we constructed a DNA replication coupled transcription and translation. So remaining reaction is a regeneration of the transcription and translation. Here, I'd like to make our goal more specific. What we need is 57 genes, encode 57 genes into DNA and regenerate them in a pure system. But this number, this number is except for ribosome. Ribosome reconstitution is, I think, still too difficult. It is, I think it should be a next challenge. So before that, we try to regenerate the 57 genes except for ribosome. Okay, so 53 genes contains two genes for DNA replication, 21 genes for tRNA, and 1 for transcription, and 33 for translation. We are now trying to reconstitute all genes, but today I show you only the result of the 15 tRNA expression and 20 translational factors expression. Okay, first tRNA. Actually, E. coli have 48 tRNA genes, but it's redundant, and the minimum requirement are 21. 20 tRNA for each amino acid, 20 amino acid, and 1 for first methionine. So we need 21. And before we started this project, already the minimum tRNA have been reconstituted. So we use that minimum codon table. And also, what we have to do is expression of the 21 tRNA in pure system. But before doing it, we encountered one problem. Pure system contaminated with tRNA. So pure system also contains the tRNA, of course, contains the tRNA. But if we didn't add tRNA into pure system still have tRNA and still have a translational activities. This is a page analysis of the protein fraction of the pure system. We found that detectable level of tRNA was contaminated in our original pure system. We are preparing pure system in our laboratory. So our lab-made pure system contained tRNA, and also commercially available pure system contained a certain level of tRNA in protein fractions. And we also found that the tRNA is contaminated in the EFTU, elongation factor TU, and ribosomal fractions. So we repurified these two proteins, EFTU and ribosome, and then we get a tRNA-free pure system. Pure system is it's tRNA-free protein fraction for pure system. Then, using this new pure system, we get no translation without the addition of tRNA, and translation only in the presence of tRNA addition, only with tRNA addition. So now we have prepared useful material, and then we try to express each tRNA from DNA in a pure system. And in this experiment, the other 20 tRNA are supplied as a purified RNA. We prepared by chemical synthesis and in vitro transcription. Then we found that, and then the tRNA activity was measured by protein expression of the reporter gene. And then we found that 15 tRNA have been functionally expressed in the pure system. And also we found that simultaneously expression of 15 tRNA also functional. So we found that at least 15 tRNAs functionally expressed in the pure system. Here we focused on only 15 tRNA because they start from G. Okay. Here we use T7 RNA polymerase. So T7 RNA polymerase can start replication, can start transcription only from guanine. Okay, so only from G. So now we succeeded. We tried the 15 tRNA expression, but we have published at this stage. But now we are trying to express the other 6 tRNA species. So, and also we have succeeded in the DNA replication coupling with the tRNA species. And also I'd like to skip this because of the time. Okay, so next is translational proteins. Here is a list of the translational proteins we have to regenerate, and about half of the proteins are ARS, amino acid tRNA synthetases. That is a enzyme that catalyzes the addition of each amino acid to a corresponding tRNA. So ARS have there is a 20 ARSs in the pure system. As a first step of regeneration, we are trying to express these ARSs in a test tube. But ARS regeneration, ARS expression is more difficult than tRNA expression because for tRNA, we don't need tRNA, okay? Because tRNA expression doesn't need tRNA. So we can remove all tRNA from pure system. There is no background. But for ARS expression, ARS needed, right? To express each ARS, we need all ARSs. So this is a difficult point for translational factors, but to assay the activity of ARSs produced in a pure system, we did a serial dilution experiment. In this experiment, first we used the complete pure system, and then that contains all 20 ARSs and expressed each ARSs from DNA. Then we diluted the reaction with a new pure system, which lacks 1 ARSs, which contains 90 ARSs. Then, if we succeeded in the expression of ARSs, then this ARS rescues the translation in the second reactions. And translation continues, ARSs continue to translate it. And also, we include this DNA for DNA polymerase. So DNA polymerase also expressed and replicated the circular DNA into a long DNAs. And we didn't show you the result, but this long DNA can be replicated by DNA polymerase. And in this experiment, this long DNA continuously replicated. So in this experiment, if the ARS is functionally expressed, then DNA concentration was kept. DNA replication continues and DNA concentration should be maintained. But if the ARS didn't express, for example, if we use GFP DNA, then DNA concentration should decrease. Using this method, we check the all activity of 20 ARSs, and we found that all the ARSs can be expressed in a functional form in a pure system. And we also performed the simultaneously replication and expression of all ARSs. We included 20 genes here, 20 DNAs here, and we used pure system that omitted all 20 ARSs. And then we found that all 20 ARS DNA continue to replicate at least 4 rounds. So that means at least 4 rounds, all ARSs can be produced. But this was published, but we found that there is a problem. The ARS is expressed, but after 4 rounds, the DNA concentration dropped significantly. And we also found that translational activity gradually decreasing. So this means ARS expression is not sufficient to keep the translational activities. So this point is the next challenge, and now we are trying to reproduce a sufficient amount of ARSs. Okay, here is a summary of my talk. This is the final slide, and what we show you today is the first Darwinian evolution of RNA self-replication system, and co-evolution occurred with parasitic RNA, and RNA spontaneously diversified, and RNA formed interdependent replication networks. And evolution still continues with a robot, because no student want to do that experiment. That experiment very laborious, but a robot can work overnight without any complaint. So I think he is very cute. Okay, so next topics is toward self-regenerative system. DNA replication was coupled with transcription translation, and Darwinian evolution also occurred. 15 out of 21 tRNA were functionally expressed in the pure system, and 20 ARSs are functionally expressed and coupled with their DNA replication. Okay, and here is acknowledgement, and he's here. This is a current program member, tRNA work by Miyachi-san here, and ARS work by Hagino-kun here. And all the DNA replication works of previous members and these members also. And DNA replication was done by Okauchi-kun. Okay, I'd like to thank them all. And thank you for your kind attention. That's all.
Thank you so much. That was fascinating and really inspiring. There is another question in chat about ribosomal RNA, I see.
Okay. Okay, can you run evolution experiment in pure for ribosomal RNA, maybe changing property of the ribosome virus? That. Yes, that is a very interesting experiment, and I want to do that, but presently we haven't succeeded. The problem is reconstitution is possible in a pure system, but only for small subunit. Small subunit can be reconstituted in pure system, but the reconstitution efficiency is low to perform the evolutionary experiment, and so we haven't succeeded yet. And also for large subunit, reconstitution of the pure large subunit in pure system have not been succeeded by anyone. So that is a technical bottleneck. Okay.
Thank you. Are there any other questions?
So may I ask question,
Yeah.
Kate?
Go ahead, Pesach. Yeah.
So this is amazing talk. I really love it. That
Thank you very much.
answer many of my question I haven't had answer for more than 50 years or whatever. So question is, I kind of related question I already asked. You show us the experiment sort of this producible, but my question is actually is like, and I'm a big fan of the movie The Matrix, so I kind of keep thinking about that movie whenever I thinking of evolution or the replication. The question is, let's say we start the same experiment or same word over and over and again, let's say 7 times. And my question is, will I exist at this time of the year, or we will have only dinosaur, or we will have the 5 arms rather than 2 arms? That was my question related to that one. Also, I comment earlier, if we have the continuously same environment, we probably don't have any diversity. We probably still have the only bacteria or even virus. That's my view of the evolution. But you demonstrate beautifully, sort of answer some of my question, but I just want to hear your perspective and the insight. regarding this kind of point, because this is a fascinating experiment. I found you today, and I regret I couldn't find you earlier. So this is amazing, amazing work. Thank you.
Okay, thank you very much for your comment. About the reproducibility, I think, yes, I show you the three independent, two partially independent experiment, and the phylogenetic tree is similar for me, and also some mutations are shared with these independent experiments, but there are many mutations unique to each experiment. So I personally think evolution is different each time. So there is a independency of the evolutions, not always the same result. That is, I think. I don't know about the dinosaur or evolution of life, but what I can say is RNA evolution have a large diversity, how to say, the large degree of freedom. So I think when we do the evolution experiment, then each time we'll get a different mutations. But maybe the phenotype may be similar. That is possibility.
Yeah, the reason I asked that question is more from the philosophical, point of view, about the fate, deterministic kind of, fate, or the stochastic result of all the factor fluctuating around me. And then that as kind of, there's no answer to that question, of course, but I just want to hear from your, your perspective. So that,
Okay,
yeah,
thank
thank
you
you so
very
much. I mean,
much.
this is amazing. I mean, I'm blown away. I mean, I want to think about this one for next 10 days or even year. I mean, thank you so much.
Thank you very much for your interesting question.
You have 3 more questions in chat.
Okay. Do you think the evolution experiment in pure would look different if you encapsulated in liposome? Okay. Would you speculate if compartmentalization change the result? Yes, we haven't done with liposomes, but I think it would be different. I think so because recently Chris Daneron's group performed the evolutionary experiment with 529 DNA replication, and they found that some mutations are liposome specific. Some mutations are beneficial only in the liposomes. So liposome-specific evolution would occur, I think. But yes, we haven't done this experiment in liposome because liposome is the encapsulating efficiency is very low. So we didn't do the same experiment using liposome. Okay, so next question. Thank you so much. Have you explored any other potential driving force complexification besides parasitism? We haven't yet. There must be some, yes, some parameters, but we don't know. We chose parasite because it's spontaneously appeared in our system and it's easy to do experiment. But if you have any other idea, I'd like to try. And other idea that can be tested, I'd like to try it. Okay. Okay. Next question. I was wondering why the pure free energy mix inhibit TDCDL. Pure free energy mixing. This system case use the pure flex 2-point energy mix. So apparent identity is not inhibited by energy mix, but by something else. Tree thought about the issue with the priming the RCA mechanism. Any thought on this? Oh, I'm sorry, I didn't catch the point. Pure free energy mix. What do you mean by pure free energy mix?
I'm sorry, it was autocorrect. I meant pure fracs.
I
Yeah.
see.
Sorry.
Pure flex. Okay, pure flex. Some mix pure flex inhibited. Yes, TTD-CDR is inhibited by NTP, high concentration of NTP and high concentration of tRNA also. So maybe both inhibited DNA replication. So apparent party not inhibited and expired. So what about the issue with primary mechanism? So if you want to do RCA in pure system, you have to reduce NTP and tRNA concentration. Is a
But sorry, I quickly switch my video. I was just wondering because the Dunwall lab was using some their catch system. They use PureFrex 2.0. And
Okay.
so they have high concentrations of DNTPs and also tRNAs, which your research, like Sakatani et al. 2015, showed that this is the trouble. So I was wondering over the last actually weeks, how is this actually possible that in the catch system, it's not inhibitory apparently, but they have a different priming mechanism because they have the protein primed. And I was just wondering whether you have any thoughts or any,
I'm sorry, I haven't used the CAD system, and I don't know where about that system. So, sorry, I don't know at this point.
That's all right. Thank you. Great talk.
Thank you very much. Thank you for asking.
I think that was all the questions. I would be very interested to see the encapsulation compartmentalization experiments done on systems like that. I think adding the individuality of a vesicle could really do some really, I don't know, maybe it wouldn't change, but that would also tell us something about the nature of encapsulation.
Yeah.
And if it does change the results, that would be super cool.
Thank you. Thank you
All
very
right.
much.
Thank you, and thanks everyone else. And have a great rest of your Monday. I hope you get some sleep tonight.
Yeah. Good night. I'll go to bed.
Bye.
Bye. Thank you very much.
Thank you
Bye.
so much.
Thanks.
Thank you. Thank you for having me.
Thanks.
Bye.
Bye.